Class 12, Chapter 12, Biotechnology and its application. Biotechnology, as you would have learned from the previous chapter, essentially deals with industrial scale production of biopharmaceutical and biological using genetically modified microbes, fungi, plant, and animals. Application of biotechnology include therapeutics, diagnostic, genetically modified crops for agriculture, processed food, bioremediation, waste treatment, and energy production. Three critical research areas for biotechnology are: first, providing the best catalyst in the form of improved organism using a microbe or pure enzyme; second, creating optimal condition through engineering for a catalyst to act; and third, downstream processing technologies to purify the protein or organic compound. Let us now learn how human beings have used biotechnology to improve the quality of human life, especially in the field of food production and health. 12.1 Biotechnology Application in Agriculture. Let us take a look at the three options that can be thought for increasing food production. First, agrochemical based agriculture. Second, organic agriculture. Third, genetically engineered crop based agriculture. The Green Revolution succeeded in tripling the food supply, but yet it was not enough to feed the growing human population. Increased yield have partly been due to the use of improved crop varieties, but mainly due to the use of better management of practices and use of agrochemicals, fertilizers, and pesticides. However, for farmers in the developing world, agrochemicals are often too expensive and further increase in yield with existing varieties and are not possible using conventional breeding. Is there any alternative path that our understanding of genetics that can show so that farmers may obtain maximum yield from their field? Is there a way to minimize the use of fertilizers and chemicals so that their harmful effect on the environment are reduced? Use of genetically modified crops is possible solution. Plant, bacteria, fungi, and animals whose genes have been altered by manipulation are called genetically modified organisms. GM plant have been useful in many ways. Genetic modification has first made crops more tolerant to abiotic stress, cold, drought, salt, heat. Second, reduce reliance on chemical pesticide, pest resistant crops. Third, help to reduce post harvest losses. Fourth, increase efficiency of mineral usage by plant. This prevents early exhaustion or fertility of soil. Fifth, enhance nutritional value of food. Example, vitamin A in rich rice. In addition to these uses, has been used to create tailor-made plan to supply alternative resources to industries in the form of starch, fuels, and pharmaceuticals. Some of the applications of biotechnology in agriculture that you will study in detail are the production of pest-resistant plant, which could decrease the amount of pesticide used. Bt toxin is produced by bacterium called Bacillus thuringiensis. Bt for short. Bt toxin gene has been cloned from the bacteria and have been expressed in plants to provide resistance to insect without the need for insecticide. In effect, created a biopesticide. Example are Bt cotton, Bt corn, rice, tomato, potato, and soybean, etc. Bt cotton. Some strains of Bacillus thuringiensis produce protein that kill certain insects such as Lepidopterans, tobacco birdworm, armyworm, coleopterans, beetles, and dipterans, flies, mosquitoes. B. thuringiensis form protein catalysts during a particular phase of their growth. These crystals contain a toxic insecticidal protein. Why does this toxin not kill this bacillus? Actually, the Bt toxin protein exists as inactive protoxin, but once an insect ingests the inactive toxin, it is converted into an active form of toxin due to the alkaline pH of the gut, which solubilizes the crystals. The activated toxin binds to the surface of midgut epithelial cell and create pores that cause cell swelling and lysis and eventually cause death of the insects. Specific Bt toxin gene were isolated from Bacillus thuringiensis and incorporated into the several crops plants such as cotton. Figure 12.1. The choice of gene depends upon the crop and the targeted pest. As most Bt cottons are insect group specific, the toxin is coded by a gene named CRY. There are a number of them. For example, the proteins encoded by the gene CRY1AC and CRY2AB control the cotton bollworm. That of CRY 1AB control corn borer. Figure 12.1 cotton ball A, destroyer of bollworm B, a fully mature cotton ball. Pest resistant plant. Several nematodes, parasites, a wide variety of plant and animal include human beings, a nematode, Melodicine incognita infect the root of tobacco plant and cause a great reduction in yield. A novel strategy was adopted to prevent this infestation, which was based on the process of RNA interference. RNA take place in all eukaryotic organisms as a method of cellular defense. This method involves silencing of specific mRNA due to the complementary double-stranded RNA molecule that binds to and prevent translation of the mRNA. Silencing the process of this complementary RNA could be from an infection by virus having RNA genome or mobile genetic element transposons that replicate via an RNA intermediate. Using agrobacterium vector, nematode specific genes were introduced into the host plant. Figure 12.2. The introduction of DNA was such that it produced both sense and antisense RNA in the host cell. These two RNA, being complementary to each other, formed a double stranded that initiate RNAi and thus silence the specific mRNA of the nematode. The consequences was that the parasite could not survive in a transgenic host expressing specific interfering RNA. The transgenic plant therefore got itself protected from the parasite. Figure 12.2. 12.2 Biotechnology application in medicine. The recombinant DNA technology process have made immense impact in the area of healthcare by enabling mass production of safe and more effective therapeutic drugs. Further, the recombinant therapeutics do not induce unwanted immunological response as in the common case of the similar products isolated from non-human resources. At present, about 30 recombinant therapeutics have been approved for human use worldwide. In India, 12 of these are presently being marketed. 12.2.1 Genetically engineered insulin. Management of adult onset diabetes is possible by taking insulin at regular time interval. What would a diabetic patient do if enough human insulin was not available? If you discuss this, you would soon realize that one would have to isolate and use insulin from other animals. Would the insulin isolated from other animals be just as effective as that secreted by the human body itself, and would it not elicit an immune response in the human body? Now imagine if bacterium were available that could make human insulin. Suddenly the whole process becomes so simple. You can easily grow a large quantity of the bacteria and make as much insulin as you need. Think about whether insulin can be orally administered or to diabetic people or not. Why? Insulin used for diabetes was earlier extracted from pancreas of slaughtered cattle and pigs. Insulin from an animal source through caused some patients to develop an allergy or some other types of reaction to the foreign protein. Insulin consists of two short polypeptide chain A and chain B, that are linked together by disulfide bridges. Figure 12.3 in mammals include human. Insulin is synthesized as prohormone like a proenzyme. The prohormone also needs to be processed before it becomes a fully mature and functional hormone, which contain an extra stretch called the C-peptide. This C-peptide is not present in the mature insulin and is removed during maturation into insulin. The main challenge for production of insulin using rDNA technology was getting insulin assembled into a mature form. In 1983, Eli Lilly, an American company, prepared two DNA sequences corresponding to A-B chains of human insulin and introduced them in plasmid of E. coli to produce insulin chain. Chain A and B were produced separately, extracted and combined by creating disulfide bond to form human insulin. 12.2.2 Gene therapy. If a person is born with a hereditary disease, can a corrective therapy be taken for such a disease? Gene therapy is an attempt to do this. Gene therapy is a collection of methods that allows correction of the gene defect that has been diagnosed in a child or embryo. Here, genes are inserted into a person's cell and tissue to treat a disease. Correction of genetic defect involve delivery of a normal gene into the individual or embryo to take over the function of and compensate for the non-functional gene. The first clinical gene therapy was given in 1990 to a 4-year-old girl with adenosine deaminase (ADA) deficiency. This enzyme is crucial for the immune system to function. The disorder is caused by the deletion of the gene for adenosine deaminase. In some children, ADA deficiency can be cured by bone marrow transplantation. In others, it can be treated by enzyme replacement therapy, in which functional ADA is given to the patient by injection. But the problem with both of these approaches is that they are not completely curative. As a first step toward gene therapy, lymphocytes from the blood of the patient are given to a cultural outside the body. A functional ADA cDNA using a retroviral vector is then introduced into these lymphocytes, which are subsequently returned to the patient. However, as these cells are not immortal, the patient requires periodic infusion of such generally engineered lymphocytes. However, if the gene isolated from bone marrow cell producing ADA is introduced into the cell at early embryonic stage, it could be a permanent cure. 12.2.3 Molecular diagnosis. You know that for effective treatment of a disease, early diagnosis and understanding its pathophysiology is very important. Using conventional method of diagnosis, serum and urine analysis, etc., early detection is not possible. Recombinant DNA technology, polymerase chain reaction, PCR, enzyme linked immunosorbent assay, ELISA, and some of the techniques that serve the purpose of the early diagnostic. Presence of pathogen, bacteria or virus, etc., is normally suspected only when the pathogen has produced a disease symptom. By this time, the concentration of pathogen is already very high in the body. However, very low concentration of the bacteria or virus at the time when the symptoms of the disease are not yet visible can be detected by amplification of their nucleic acid by PCR. Can you explain how PCR can detect very low amount of DNA? PCR is now routinely used to detect HIV in suspected AIDS patient. It is being used to detect mutation in genes in suspected cancer patient too. It is a powerful technique to identify many other genetic disorders. A single standard DNA or RNA tagged with a radioactive molecule probe is allowed to hybridize to its complementary DNA in a clone of cells, followed by detection using autoradiography. The clone having the mutated gene will hence not appear on the photographic film because probe will not have complementarity with the mutated gene. ELISA is based on the principle of antibody interaction. Infection by pathogen can be detected by the presence of antigens, protein, glycoproteins, etc., or by detecting the antibodies synthesizing against the pathogen. 12.3 Transgenic animals that have had their DNA manipulated to process and express an extra foreign gene are known as transgenic animals. Transgenic rats, rabbits, pigs, sheep, cows, and fish have been produced, although over 95% of all existing transgenic animals are mice. Why are these animals being produced? How can man benefit from such modifications? Let us try to explore some of the common reasons. First, normal physiology and development. Transgenic animals can be specifically designed to allow the study of how genes are regulated and how they affect the normal function of the body and its development. Example, study of complex factor involved in growth such as insulin-like growth factor by introducing genes from other species that alter the formation of this factor and studying the biological effect that result. Information is obtained about the biological role of the factor in the body. Second, study of disease. Many transgenic animals are designed to increase our understanding of how genes contribute to the development of disease. These are specifically made to serve as models for human disease so that to investigate of new treatment for disease is made possible. Today, transgenic models exist for many human diseases such as cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer's. Biological products. Medicines required to treat certain human disease can obtain biological products, but such products are often expensive to make. Transgenic animals that produce useful biological product can be created by the introduction of the portion of DNA or genes which code for a particular product such as human protein alpha 1 antitrypsin used to treat emphysema. Similar attempts are being made for treatment of phenylketonuria (PKU) and cystic fibrosis. In 1997, the first transgenic cow, Rosie, produced human protein enriched milk 2.4 gram per liter. The milk contained the human alpha lactalbumin and was nutritionally a more balanced product for human babies than natural cow milk. Vaccine safety. Transgenic mice are being developed for use in testing the safety of vaccine before they are used on humans. Transgenic mice are being used to test the safety of polio vaccine. If successful and found to be reliable, they could replace the use of monkeys to test the safety of batches of the vaccine. Chemical safety testing. This is known as the toxicity or safety testing. The procedure is the same as that used for testing toxicity of drugs. Transgenic animals are made that carry genes which make them more sensitive to toxic substances than non-transgenic animals. They are then exposed to the toxic substance and the effects study. Toxicity testing in such animals will allow us to obtain result in less time. 12.4 Ethical issues. The manipulation of living organisms by the human race cannot go on any further without regulation. Some ethical standards are required to evaluate the morality of all human activities that might help or harm living organisms. Going beyond the morality of such issues, the biological significance of such things is also important. Genetic modification of organisms can have unpredictable results when such organisms are introduced into the ecosystem. Therefore, the Indian government has set up the organizations such as GEAC, Genetic Engineering Approval Committee, which will make decisions regarding the validity of GM research and the safety of introducing GM organisms for public service. The modification or usage of living organisms for public service as food and medicine source, for example, has also created problems with patient guaranteed for the same. There is growing public anger that certain companies are being guaranteed patent for products and technologies that make use of the genetic material, plant, and other biological resources that have long been identified, developed, and used by farmers and indigenous people of a specific region or country. Rice is an important food grain, the presence of which goes back thousands of years in Asian agriculture history. There are an estimate 2 lakh varieties of rice in India alone. The diversity of rice in India is one of the richest in the world. Basmati rice is distinct for its unique aroma and flavor of and 27 documented varieties of basmati are grown in India. There is reference to basmati in ancient texts, folklore, and poetry as it has been grown for centuries. In 1997, an American company got patent rights on basmati rice through the US Patent and Trademark Office. This allowed the company to sell a new variety of basmati in the US and abroad. The new variety of basmati had actually been arrived from Indian farmer varieties. Indian basmati was crossed with semi-dwarf varieties and claimed as an invention or a novelty. The patent extends to functional equivalent, implying that other people selling basmati rice could be restricted by the patent. Several attempts have been made to patent uses product and processes based on Indian agriculture, herbal medicine, example turmeric, neem. If we are not vigilant and we do not immediately counter these patent applications, other countries or individuals may encash on our rich legacy and we may not be able to do anything about it. Biopiracy is the term used to refer to the use of bioresources by many multinational companies and other organizations without proper authorization from the countries and people concerned without compensatory payment. Most of the industrialized nations are rich financially but poor in biodiversity and traditional knowledge. In contrast, the developing and the underdeveloped world is rich in biodiversity and traditional knowledge related to bioresources. Traditional knowledge related to bioresources can be exploited to develop modern applications and can also be used to save time, effort, and expenditure during their commercialization. There has been growing realization of the injustice, inadequate compensation, and benefit sharing between developed and developing countries. Therefore, some nations are developing laws to prevent such unauthorized exploitation of their bioresources and traditional knowledge. The Indian Parliament has recently cleared the Second Amendment of the Indian Patent Bill that takes such issues into consideration, including patent terms, emergency provision, and research and development initiation. Summary: biotechnology has given the humans several useful products by using microbes, plant, animal, and their metabolic machinery. Recombinant DNA technology has made it possible to engineer microbes, plant, and animals such that they have novel capabilities. Genetically modified organisms have been created by using methods other than natural methods to transfer one or more gene from one organism to another, generally using techniques such as recombinant DNA technology. GM plant have been useful in increasing crop yield, reduce post-harvest loss, and make crop more tolerant of stresses. There are several GM crop plant with improved nutritional value of foods and reduce the reliance on chemical pesticide. Pest resistant crop recombinant DNA technology processing have made immense impact in the area of healthcare by enabling mass production of these safe and more effective therapeutics. Since the recombinant therapeutics are identical to human protein, they do not induce unwanted immunological response and are free from risk of injection, as was observed in case of similar products isolated from non-human sources. Human insulin is made in bacteria, yet its structure is absolutely identical to that of the natural molecule. Transgenic animals are also used to understand how genes contribute to the development of a disease by serving as models for human disease such as cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer. Gene therapy is the insertion of genes into an individual cell and tissue to treat disease, especially hereditary disease. It does so by replacing a defective mutant allele with a functional one, or gene targeting, which involves gene amplification. Viruses that attack their host and introduce their genetic material into the host cell as part of their replication cycles are used as vector to transfer healthy genes, or more recently portion of genes. The current interest in the manipulation of microbes, plants, and animals has raised serious ethical questions.